No?
We can
Okay.
start now. It is
Okay,
16:10. I have the honor to welcome you to a scientific seminar dedicated to the fascinating topic Feeling the Force, topics and technique to unravel the cellular forces. The seminar will be presented by Dr. Sam Aytekin. I'm correct?
Not yet doctor, though, but I'm on the way.
Okay. Sam Aytekin is a doctoral researcher.
Yes.
In a Roche lab, molecular imaging and photonic research unit, Department of Chemistry, University of Leuven.
Yes, in Belgium.
Belgium. He is expert in a field of cellular biology and extensive experience in studying cellular forces and their impact on biological processes. Sam Aytekin will introduce you to the latest tools and methods that allow us to sense and unravel cellular forces. You will learn about the recent advancement in this field and their potential impact on understanding of cellular biology. Please join us and get ready for an exciting presentation of by Sam Aytekin. Please, Sam, you may begin.
Thank you very much for the very nice introduction. Actually, you already summarized the topic way better than I could do. So maybe I can try to share my screen and then we see if you guys see my slide properly.
Yeah, all fine.
Yeah.
All fine, Tony. Okay, thank you very much. So of course, I also need to start by thanking all of you a lot, specifically my dear friend Tatiana Zakisulu and Professor Monastirsky, and of course also Kyiv Polytechnic Institute. So this is a bit of an exciting talk for me and also a bit stressful, to be honest, because this is the first time I am giving a talk to an audience that is not necessarily from the same background that I have. So I had to simplify things. I needed to keep things very, very basic, and I noticed that explaining science in simple terms is not the easiest thing to do. I would like to have this disclaimer that I am just a second-year PhD student, so I'm absolutely not an expert in this topic. I'm not a professor with decades of experience on it, but I'm going to try to do my best. And at the end of the day, the idea is to have a bit of fun and learn one thing or new thing so we can move on. I would like to start my presentation with a couple of videos. So what we see here is a cell passing by, and when it's passing, you can see that it's actively densifying the collagen fibers around it. So by doing so, it's creating a stiffer environment around the cell, which lets it to attach it better, to move better. Here in this video, we see that again, two cells, they are embedded in the collagen matrix and they are remodeling, they are deforming the collagen fibers around them to form this kind of bridge between them so that they can reach to each other. And this video is my absolute favorite in biology. We see a cancer-associated fibroblast, a single cancer-associated fibroblast carrying an entire mass of cancer tissue, a tumor tissue with it. So you can already imagine how much forces that it can be applying there. So these are just a few examples to illustrate the physical forces between the cell and its environment. Let it be extracellular matrix, let it be a different cell, has an immense effect on the cell behavior. So I just mentioned extracellular matrix, but what is it really? So I'm just hoping that you actually heard these concepts, but I can't be sure because I know there are some physics students among us too. So I would like to really try to explain all the basic concepts too. So just like you guys are surrounded by air, which is made up of oxygen, nitrogen, the dust particles, the perfume, and the deodorant, the cells are also surrounded by a substrate. And this is what we call extracellular matrix or ECM. And it's a quite complex structure, actually, structural-wise, composition-wise, architectural-wise, and mechanical-wise. Extracellular matrix is very complex. So the main ingredient in extracellular matrix is the collagen fibers, so that's good. But there's also hundreds of more proteins out there. It can be fibronectin, laminin, elastin, growth factors, other cell types, many things. So practically speaking, it's almost impossible to recreate extracellular matrix in the laboratory. But what we can try to do is to mimic it. So as I said, it's like 90% collagen fibers. So what we can do is to use the collagen as our substrate. We can, of course, mix always collagen, fibronectin, laminin to somehow mimic the extracellular matrix too. Or we can just go with the synthetic hydrogels, for instance, to mimic maybe not the mechanical properties of it, but maybe the structural properties, the porosity and the length of the fibers and so on, or simply the stiffness. But then the question is, why would we even bother to mimic the extracellular matrix. So when you think about it, the biologists for such a long time, we believe that it's the genes, it's the DNA, it's the proteins, it's the signaling pathways that are regulating the life. But in last 2 decades, we came to terms to understand that it's actually the mechanical inputs and the physical forces are as much important as the biological cues for cellular behavior. In fact, that is responsible for the most basic one, most fundamental cellular behavior, such as proliferation or the division of one cell into two. Nowadays, scientists think that the cell is doing it not because to increase its number, but also reduce the mechanical stress over itself. Another example can be the migration or the motility of the cells. So the cells can move around within seconds, or they can stand still for days, just depending on the architectural and mechanical properties of their surrounding. The next one is differentiation, which is very important. So as you know, when we start our life as an embryo, we start as a single cell organism, and then we divide, and then we become 2 cells, and then 4 cells, and 8, and then 16. But if we just keep dividing like this, I think at the end of the day, we will just end up being a blob of cells, and I bet that wouldn't look very beautiful. Instead, what happens is in the early stages of the development, these cells, even though they are exactly the same at the beginning, start to differentiate into different cell types. They form brain cells, they form bone cells, they form heart tissue cells. So eventually they make us. And this is the mechanical input there is very important, which you can something very easily check. All you need to do is to take a stem cell and then put it in 2 different substrates with 2 different stiffnesses, and you are going to see that the stem cell differentiates or forms into 2 different cell types. So I think it's very cool. But the real deal there, why so many biologists nowadays are interested in this mechanical input on the cell behavior, is because its effect on the pathological conditions. So I already showed you one cancer-associated fibroblast is pulling the entire mass of the cancer cell. So you can immediately think of metastasis, the final stage of cancer. So when the cancer cells in your body leave their bed and start to travel around in your body, that's bad news. And the same thing goes for the angiogenesis too, the formation of new blood vessels around the cancer cell. So the mechanical properties of the surrounding is very important for this behavior. Or you can take any tissue, and if the stiffness of this tissue increases, that is going to activate what we call fibroblast cells, which are going to interdensify the collagen fibers around them to the point that the tissue is scarred and it's not working anymore. So at the end of the day, what you end up with is a lung fibrosis, is a liver cirrhosis, or is a atherosclerosis. So, but then the question is, how do we solve these problems? How do we prevent these diseases happening, or how can we treat them? Of course, the answer is a bit simple. It's by understanding it, understanding this phenomena. So how we are going to do that though? I asked ChatGTP to tell me what is mechanobiology, and this is what it gave me. So mechanobiology seems that is an interdisciplinary field that explores the ways that the cells sense and also responds to mechanical forces in their environment. As I said, it can be extracellular matrix, it can be other cells, it can be just the substrate that you are using to grow the cells on. With the mechanotransduction being the process through which these forces, external forces, are converted into biochemical inputs so that it determines the fate of the cell. So let's talk a bit about mechanotransduction now. So imagine you have a cell here, which is extremely simplified version how a cell looks like, and there is another cell there. So the forces can be created between the 2 cells in cell-cell junctions, or between the cell and the substrate that is lying or the substrate that is growing inside. So once these forces are created, that is sensed by what we call like force-sensitive proteins. They are usually in the focal adhesions or in the cell-cell junctions. So it can be cadaverine, integrin, taurine, vinculin. And these proteins basically sense the force. And depending on the force, they go through these conformational changes. Once it's done, the force is transmitted to the cytoskeleton of the cell. So this is just like our skeleton. It's like the lines, the columns are reaching all over the cell. Here is depicted in red, and it's reaching to the nuclear membrane. Remember, nucleus is where the DNA is stored, right? So in the nuclear envelope, there are also force-sensitive proteins that tells nucleus, okay, activate this gene, deactivate this gene, and depending on this gene activation or gene regulation, you have an output, a cellular response. It can be, again, proliferation, it can be differentiation, it can be cell death, it can be practically anything. But I want to also show how these important players in mechanotransduction look like under microscopy. So we can start from the cell-cell junctions. This is how it looks like. The green lines that you see are basically the junctions between two cells. And the most common protein in cell-cell junctions are the cadarins. So in fact, what we see here is labeled cadarin proteins. Just one note, maybe not every cell forms cell-cell junctions. For instance, the endothelial cells, the cells surrounding our blood vessels, they form cell-cell junctions so that the blood doesn't leak, of course. Or our epithelial cells forming the skin layer, they form these strong cell-cell junctions. So of course, for us, it's very interesting what happens to these forces in cell-cell junctions if we, for instance, apply interstitial flow, if we apply a shear stress of them. What is the integrity between them? So it will be very cool if we could measure the forces between a cell and another cell. The next one, the focal adhesions, they exist in almost every cell type. So focal adhesions, what we see here is a kind of purple spheres. You can imagine them as arms of the cell, and they compose of many proteins. The most prominent ones are the integrins, taline, vinculin, actinin, and so on. And you can imagine these proteins as the fingers of the cell. So focal adhesions are responsible for cell to attach their environment and to pull their environment so that they can move around. So it's really acting like an arm and a finger. And here in this image, all these yellow dot kind of structures are the focal adhesions, the fingers of the cells. The next is the cytoskeleton. As I said, just like we have our skeleton in our body, cytoskeleton is important for cell shape, to contain its shape. And here the yellow lines are the actin filaments. They are really going from one end of the membrane to the other end of the membrane, or they can connect two different focal adhesions to each other. They can connect one focal adhesion to the nuclear membrane. They can do many things, but in this concept, mainly they are important for the transmission of the forces from outside to the inside of the cell. And the last one is our final destination for the forces, the nuclear membrane. So here you can see it as green circles. By the way, I have a question. So in my screen, I see all of you in the right side of the screen. Do you also see it that way, or do you see just my full slides?
We see your full slide.
Perfect. So okay, because in my case, the Zoom is kind of blocking the way for the slide, but it's fine for me. Yeah, thank you. So if you see it nice, it's fine. So okay, so I hope I kind of made it clear that it is important to understand these processes, but in order to be able to do that, we need to find a way, we need to be able to measure these cellular forces in our laboratories. And today's topic will basically focus on that. So there are many ways of doing that, measuring these forces, but two of the methods are really outshining the others. The first one is the traction force microscopy. This is quite a standard technique in mechanobiology. And the other one is more recent development, but it's an extremely powerful technique to measure the forces, and that is what we call molecular tension probes. So let's start with the traction force microscopy. TFM, in very simple terms, measures the deformations in your substrate, and from these deformations, it calculates the tractions or the cellular forces. So one of the earliest examples of TFM is using these wrinkle studies. What you do is you take a cell, you put it on a substrate, an elastic substrate that can wrinkle as a result as the cell is like contracting each other. And then you get this kind of deformations. And from there, you try to calculate how much force the cell might have applied to create this much of wrinkle. But yeah, as you can already imagine, that resolution might be horrible here. Like there is this one giant wrinkle associated with one side of the cell. So it's just one force, one edge of the cell. It's really not good. Also, the substrate selection will be very bad in this case because you need to find a material that is going to form wrinkles as a result of piconewton forces. So you're a bit limited there too. So this is what we call like a peasant way of doing the science. And I really hope none of you are working on that. So the next development is actually using micropillar based TFM. So here we have these PDMS based micropillars. They're like, their diameter is usually around like 1 micrometer, which is actually quite large for biological purposes. but fine. So there the cell is sitting on top of this pillar, and the stiffness of the substrate you can easily change by changing the length of the micropillars. And then by looking at how much these micropillars are pulled, you can calculate the deformation, and from that deformation, you can move to the tractions. But then again, inside our body, the cells are not really lying on top of the pillars, right? So they are in direct contact in everywhere in all three directions. So the better way of doing TFM is using bead-based one. So in this case, we are using a hydrogel, an elastic or at least semi-elastic hydrogel. The problem is we cannot follow the deformations of the hydrogen fibers. We can't even see it in our confocal microscopy. So instead, what we do is to put fluorescent beads inside the hydrogel when we are preparing it. So whenever the cell moves around and pulls the fibers of the hydrogel, these fluorescent beads inside also move. So from there, we can measure the deformation of, deformation of the fibers, but in this case, the movement of the beads, and from the movement of the beads, we can move to the tractions. So it looks like this when it's illustrated, but in reality, it looks like this, of course. So here you see a cell is moving around, and with every movement of the cell, you see that the beads is also moving around that. So if our algorithm can follow the movement of these beads, we can from there calculate the tractions. So this is how experimentally TFM looks like. It's relatively simple experimental parts. We start with a hydrogel. We can prepare it within an hour in our laboratories. We embed our beads in it. Depending on your choice, you can either put your cells on top of the hydrogel or inside of the hydrogel. So if you are interested in 2D TFM or if you are interested in 3D TFM. And then, okay, you let like 1 day or a couple of hours at least for cells to spread. As they are spreading, as I said, they are pulling the fibers towards themselves. And with this movement, the beads are coming towards themselves too. Then you take a image of the beads per se, and this is what we call stress state because the fibers are in stress state. So the cell is spread. Then we treat the cells with a sort of relaxing agent, or we can simply kill them if we are not interested in them afterwards. So the cell is gone, cell is not applying forces anymore. And remember, this is an elastic substrate, which means the fibers will go back to their original position. Then we take another image of the beads, which is what we call relaxed state beads. And all we need to do afterwards to compare these two images to each other, see how much the beads move, and from there, by doing some pre-processing, we calculate the displacement fields, and from the displacement fields, we move to the tractions or the cellular forces. I wanted to talk quickly about the substrate that we can use. So the most common way of doing bead-based TFM is using polyacrylamide hydrogels. This is very easy. All we need to do is to mix the acrylamide and bisacrylamide. And just by changing the ratio between this acrylamide and bisacrylamide, you can end up having different hydrogels with different pore size or different stiffnesses. So this one is easy to do. Another good property of that is it's elastic. As I said, like we really want a material that will let the beads in it to go back to their original position. And maybe the most important that this material is linear material. So linear material means there's a linear relationship between the stress and the strain. So in this case, we are looking at the strain, the deformation, and from this deformation, we are trying to calculate the stress or the forces that the cell applied there. So in order to go from displacement to traction, we really prefer to have a linear material. If we move to a nonlinear material, for instance, most of the natural hydrogels are nonlinear, then it's a bit tricky because with every stress applied on the material, stress-strain ratio is also changing. And nowadays, many people are trying to solve this issue to make nonlinear material be usable for TFM purposes, but it's still a question if we can do it or not, or I mean, we can do it, but can we trust the traction values that we get afterwards or not? So I know people are working on it, but most of the time people prefer to stop after the displacement measurement. So at the end of the day, actually what you do is not traction force microscopy, it is displacement microscopy. So you can still have a look at, okay, in which side of the cell there is more displacement, where there are more forces applied, but you cannot really give values of how much pascals or how much piconewton forces are applied there. So, okay, that's out of the way. Experimentally speaking, things look like this. Here we have an intensity image of a cell, and all these bright dots are the focal adhesions that I showed you. And then we have a stress state beats image, and then we have a relaxed state beat image. Then all we need to do is to combine them and analyze them. So every yellow dot that you see here are the beats that didn't move. So they were in the same location between the stress and the relaxed states. But here in these three regions, you can actually differentiate between the red and green beats, which means the beats move there. So from there on, we just calculate the displacement in x-axis and then y-axis, and then we put them together to calculate this displacement field, which is given by micrometers here, as you can see. So every red region here moved approximately 1.6 micrometers. And from there on, if we have a linear material, we can easily reach to the tractions associated with it, which is given in pascals. So this is very cool. We can really see which focal adhesion is applying more forces and which focal adhesion is applying less forces. We can even have values associated with it. So for the fundamental science purposes, this is amazing, of course. I just wanted to quickly talk about the formula part of it. Well, I'm really not very good at it, but if you are doing 2D TFM, we are usually using Green's functions, which is taking the displacement and is converting it to the forces. But the Green's functions assumes this semi-infinite half space. So it's assuming this like infinite XY plane and infinite in one direction in Z plane, which is of course not the case for our hydrogels. So there are a lot of formulas going on there, a lot of assumptions are going on there. So it's not as simple as it looks. For the 3D TFM, we cannot even use the Green's formula because there is no semi-infinite space anymore. The cells are surrounded by your hydrogel in every direction. So what we do is to use finite element method. I'm pretty sure you guys know this much better than I do. But basically what we do is dividing one big problem into smaller problems. We are creating these meshes, both for the cell and for the hydrogel. And by looking at how much these meshes move and comparing it to the neighboring mesh, we calculate the displacements. And again, if you have a linear material, you can go to tractions from there on. But the only reason actually I put this slides, I was hoping that there will be many young physics students out there, and I was going to say that we biologists, we need you. I personally work on TFM, but even these formulations, the software developments and this and that, I do not understand. I just do my experiment, I analyze it, then I go to a physics department or a engineering department to ask them, look, do results, do they make sense? So for the young generation, we actually need you. We need some people to work on our algorithms, work on our modelings, work on this software and help us analyzing these kind of things. So that's why actually I really wanted to give this speech to you, because I know this is an applied physics department as far as I know, and I know biology may not be that relevant to you, but there are still parts that you can take parts of. That's out of the way, and I feel a bit ashamed now. We can move to the second method, which is the molecular tension probes. So there are many different versions out there, but one of them is really what everybody uses, to be honest. I mean, no one then someone comes up with a different idea, but then there is this one of them that is already optimized nicely, characterized nicely, and it works nicely. So usually we stick to this one. But almost all of them uses a phenomena called FRET, or Forster resonance energy transfer. And before explaining how these molecular tension probes work, I think I need to explain how the FRET works. So FRET is a phenomena where the energy of the electron is transmitted to another molecule as long as this molecule is close enough to the first molecule. So if you look at this video, when you excite a donor molecule, a fluorescent molecule, is going to emit a light, but nothing is going to happen to the next molecule unless they are closer to each other, less than 10 nanometers. When it happens, when you excite a light on a fluorescent molecule, it's going to transfer its energy to the next fluorescent molecule, which will be excited, and when it's going back to the ground state, it's going to emit a light. So it's a strictly distance-dependent phenomenon. I'm going to explain in more physicist terms. I will try my best. So first of all, okay, we call it donor and acceptor, but both of them are fluorescent proteins. It can, sorry, fluorescent molecules. It can be a protein or it can be a dye. So if it is inside of the cell, we are using fluorescent proteins. If it is outside of the cell, we are usually using fluorescent dyes. For this FRET phenomena to happen, one of the criteria is that we need to have a spectral overlap between the emission of the donor and the excitation of the acceptor. So when we say this, usually people tend to think like, okay, when the donor emits a light, this light excites the acceptor, but it's not true actually. There is no light transfer between the two molecules. There is an energy transfer. So if you look at this diagram, so usually when you excite the electrons in a fluorophore, when it's going back to the ground state, it emits a light. This is like one of the probabilities. How do you go back to the ground state? How do you release your energy? And with the fluorescent proteins, the most common one is what we call fluorescence. But if there is another fluorescent molecule with a spectral overlap that is closer, less than 10 nanometers to this first fluorophore, then we add yet another probability to how to lose this energy. So the donor molecule may transfer its energy to the next fluorescence, and then it's going to be excited, and when it's going back to the ground state, it's going to emit a light at different wavelength, of course. So this is, as I said, extremely distance-dependent phenomena. It's just Armstrongs we are talking about. It's just less than 10 nanometers, and this is so good for us because the biological phenomena happen in this length scale. So most protein-protein interactions, whatever you can think about biology, I can assure you is going to be within a couple of nanometers. So we can use FRET phenomena for our advantage then, and we do indeed biology. We do use FRET practically for everything. All you need is a microscope and a fluorescent proteins, two sets of them, and then you can study protein-protein interactions, how the enzymes are activated or deactivated, how enzymes actually work. So we really use it for everything. But in today's topic, we want to use it for force calculations, right? Or tension calculations. So how can we do that really? So we want a system that is going to change its distance or length as a result of an applied force. So we can think of something like a spring, right? If you apply a tension to a spring, it is going to open up. And we know that FRET efficiency is going to change depending on how my donor and acceptor are close to each other. So if you are applying a tension, let's move to the next slide. This donor and acceptor molecule will be separated apart from each other, which means there will be no FRET happening anymore. And if you are not applying any tension, the spring will go back and the donor and acceptor will be very close to each other, and then you will have a higher FRET efficiency. Okay, but how can we apply a tension in this kind of sensor? So all we need to do is to take this sensor and put it in a force-sensitive proteins that I mentioned beforehand. So it will look like this now. We will just insert it into a protein. And then whenever there is a tension applied in this protein, the FRET efficiency will decrease. So this is something that we need to remember all the time. Higher FRET efficiency, lower tension, lower FRET efficiency, higher tension. Okay, so the first one that is developed was for the winkling, actually. So I would like to zoom in to focal adhesions to explain it a bit better. So this is what we see here is a focal adhesion. So it's, as I said, composed of integrins, taline proteins that is connecting integrin to the actin filaments, vinculins that are connecting taline proteins to the actin filaments. And we are mostly interested in the vinculin because it's such a good model protein that we can work on the forces. So if we zoom in there, vinculin has a head domain that is attached to taline and a tail domain that is attached to actin, and then everything else in between. So if we put our force sensor here, as the integrin is pulling from this side, and as the actin is pulling from the other side, my donor and the acceptor will separate apart from each other, and you will see a decrease of FRET efficiency. So again, you will see a decrease of FRET efficiency, which indicates these molecules are in higher tension. But in biology, it's very important to have a control group because the cells doesn't have a mouth and they cannot tell you what is going on there. So if you want to know your sensors are working, you need a good control. And in this case, this is how we do it. We basically remove the tail domain of the vinclin, which means one end of the spring is free floating now. So it doesn't matter how much tension the integrin applies from here, this sensor is not going to open because it's not attached in two sides. So it will always give me higher fret efficiency, which means lower tension. Well, in this case, it's not force sensitive, so it's tensionless. Okay, so we can do many things about it. We can first of all choose our favorite fret pair. Imagine you have a fluorescent microscopy and your lasers are not fit to excite the cyan and yellow fret pair, then you can simply switch to the green and red fret pair. Also, you can change the elastic linker in between. So in the original molecular sensors, they were using this helical structure polypeptide, which is actually obtained from spider silk. But now we kind of replaced it with an unstructured polypeptide because we saw that it has a much better sensitivity to resolve the forces or resolve the fret efficiencies. But still, if we look at these images, we can really see that for a given focal adhesion, within just a single focal adhesion, we have a big change of fret efficiency, which means there is a big change of tension there. And if you look at the colors, this is fret efficiency color bar. The blue part means lower fret efficiency, which means higher tension that we see in the cell edges. And the more red spectrum means that higher fret efficiency, lower forces or lower tensions inside the cell. So we didn't stop with the vinculin. Of course, I put it also in other force sensitive proteins. For instance, I put it in cadaverine sensors. So in the cell-cell junctions, now we can actually measure what is going on there, which kind of forces are there. But also I put it in the Nesperin. Nesperin is a nuclear envelope protein, and that's basically where the forces reaches at the end. And there we can also see that now how much average force or average tension is applied in the nuclear membrane. Of course, if you are just looking at one one cell, I understand that you might be thinking like, why would I even want to learn what is the force in a nuclear membrane? But of course, these techniques are to compare two different scenarios, right? For instance, comparing a pathological condition with a healthy condition to understand like why active fibroblast is acting different from inactive fibroblast. Is the forces in the cell membrane different? Is the forces in the nuclear membrane different? So this is really check all of this stuff. Before closing this chapter though, I want to mention something. So if you notice, I wasn't talking about the values of the tractions or values of the forces. I never give you piconewtons and this and that, because to go from fret efficiency to piconewtons, we need calibration curves. But how these calibration curves are done is basically like this. You take this sensor out of the cell, you basically attach it to a surface in one end, so it's stable there. And in the other end, you are using optical tweezers, you use a magnetic bead, and you know how much force that you are applying, and then you are basically measuring the fret efficiency between donor and an acceptor. And then you make this calibration curve, fret efficiency versus force. Another way of doing it is using some models. So you use experiments to get a couple of parameters out, and then you go ahead with this worm-like chain model, and you insert these parameters in, and you try to model it. And again, you end up with this calibration curve of fret efficiency versus force. But the problem is none of these models or the approaches are done inside of the cell. So we are obtaining these calibrations either like outside of the cell in vitro or in silico. And this is biology, right? We cannot know what is going on inside the cell. Even two cells next to each other, they can act very different from each other. So it's not like CMOS technology that you use a machine and create all these exactly same looking transmitters, I think you call it. It's very, very different. We don't know what is the pH inside the cell. We don't know what kind of reactive oxygen species around these molecules. So then the question is, can we actually talk about piconewtons when we are using these molecular tension probes? Usually we prefer to stop at fret efficiency. Okay, we look at the fret efficiencies and we say that this particular focal adhesion applied more force or less force compared to this particular focal adhesion. But I think it's not really accurate or precise to talk about the forces there. So still we need to optimize that process. But anyways, it's working and we get these beautiful looking cells. Again, the blue parts representing the tension, higher tension regions, and the more red or yellow parts are representing the lower tension regions. And if we zoom in one of them, we can really see over one focal adhesion, we go from higher tension towards the lower tension. So this is very, very powerful technique, because when you think about it, you don't need any substrate, you don't need any external material, you don't need algorithms, software, some good computer that can analyze things for you. All you need to do is to transfect yourself or make yourselves express these sensors, and the cell is going to tell you what is happening inside. And of course, we also need to think about the resolution. When we are talking about TFM, we are looking at the beats, literally micrometer sized beats, and how much do they move. So usually when we talk about the resolution of forces in TFM, we are talking about like 5 micrometers. These sensors are molecular sensors. They are actually telling us what is going on in the single molecular level. So if I need to use an analogy, for instance, I can lift my laptop, right? And you can measure how much force that I applied on the laptop. But then how much force my fingers applied. You can try to estimate it by dividing the total force with the number of my fingers, and hopefully I have 5 of them. But then again, you are still not knowing which finger is applied how much force. There is literally no way of doing it with the TFM. But the molecular sensors, each finger is a sensor. So they can tell you under how much tension I am in. So that also concludes that chapter. And this is the last chapter of mine. I think I'm going to take like 5 more minutes maybe, and then I will be done. So far we were talking about outside of the cell by using traction force microscopy, by looking at the deformation on a substrate, and inside of the cell by using the molecular tension probes. But what if you are interested in outside of the cell, but in the molecular level again? So how can we increase the resolution and we get much better than traction force microscopy? Then, okay, outside of the cell, we cannot use the proteins. Proteins are for the inside of the cell for many reasons. And also we cannot use fluorescent proteins, which means we need to use fluorescent dyes. But what we can use is actually DNA. So what we are looking at now are the integrins this time, and we are looking at outside of the cell. Now we are not inside of it. So this you can imagine like an extracellular matrix, or you can imagine like a substrate. So we can use DNA instead. What we see here is a DNA hairpin loop structure. This is a single DNA strand. Then we have a second strand here and a third strand here. So the second strand is carrying a donor fluorescent dye, and the third strand is carrying a quencher dye. So in the fluorescent protein-based thread, remember, donor was helping acceptor to emit a light. In this case, if they are closer than 10 nanometers, quencher quenches all the signal of donor and doesn't emit anything. So this is the configuration when the integrins or the cells do not apply any force on it. But if the integrins pull the substrates, then the hairpin structure is going to open, donor and the quencher will go apart from each other, which means when you excite the donor, now you can get an emitted signal. So this is very cool, because just by looking at the presence or absence of a signal, we can actually say that how much force the cell applied there. So we can actually simply change the GC content, so guanine cytosine content of the DNA, to end up having different DNA force sensors that opens at different opening forces. So if it is 22% GC, for instance, it's going to open at around 4.2 piconewtons, which you can see here. One thing to notice though, unlike the molecular tension probes, these are digital systems. So DNA hairpin is either open or close. So if you are getting a signal, rest assured that you are having a force that is more than that is opening threshold force. So I found this topic very, very exciting. I wish we would have figures that also looks very exciting, but it's a bit hard to get with the DNA force sensors. They usually look like this and people are like, what do we even see there? You see a signal. And as I said, if you have a signal, that means a part of like a particular force is applied there. And if you are using if you mix different DNA force sensors at different opening forces together, and if you functionalize your substrate or surface with them, you can really see at what part of the cell, okay, 5 piconewton or more force is applied. At what part of the cell, 8 piconewton force or more is applied. You can do very different things with it. You can create DNA origami structures, and you can, put different DNA force sensors on it that opens at different threshold forces. There is this group who made these DNA force sensors, not only giving you the forces, but also giving you the orientation of the forces, which I do not understand how, but I found it very cool. And then another way of doing it is this memory kind of structures. So all you need is a locking strand and unlocking strand in this case. So you can actually remember which DNA force sensors were open at what point of time. And my last slides, we can do super resolution microscopy with that too. I'm not going to go deep into DNA paint super resolution microscopy, but if you are thinking that this figure looks very blurry, I want to invite you to have a look at this scale. So if anybody among you work with the fluorescent microscope or confocal microscopy, you know that our resolution limits because of the diffraction is around 200 nanometers. We cannot go beyond that. And here, every single signal that you see is already like 20 to 30 nanometers. So our resolution is really, really good. We can actually see the opening of single DNAs in this image, and which you can easily compare here with a diffraction limited microscopy with a DNA paint-based DNA force sensors. So there are many things to unravel there, but I think my time is a bit up, and I would like to stop it here. I added these slides for acknowledgement, but I don't want to talk about my group right now. I just wanted to tell you, please go ahead and ask your questions. You can always contact me or my professor, my supervisors. You can always find us in the website of Russia Lab 2. And yeah, this image is actually from last Christmas party. And yeah, Christmas is approaching, and I want to actually celebrate your Christmas too, because one of my favorite Christmas song, and actually one of my favorite songs, Christmas Carol, Tatiana told me that it is actually composed in Ukraine. So I'm proud of you guys. And yeah, that's it. Thank you very much for listening to me. Now shoot.
Thank you, Sam.
Thank you, Sergey. You hear me or not?
Yes, of course. Yes, of course, I hear you.
First of all, I would like to say many thanks, Samet, and I'm sorry I have a small transport trouble.
Oh, it's no problem at all. You were there in time, no problem.
But I saw that Sergey was very good in role of seminar, and Sergey, please continue in.
Okay.
Continue.
Okay. Thank you, Sam, for a terrific presentation. Colleagues, if you have any questions, please ask.
Yes.
I can ask general.
May I ask?
Okay. Then I'll generate.
Yes. As we know, and as you have shown for us, that moving in the cells participate a lot of different kind of proteins.
Yes.
And if you look inside the cell, how you think what governs the sequence of the events?
Yes, I understand.
If you're considering the body as whole, it's the brain and nervous system governs all events, and in the cell, what governs this sequence of events?
Indeed. So the end results of a cell behavior doesn't matter if it is neuron or stomach cells or anything. Is really that it comes to the gene expression and of course protein-protein interactions, which is regulated by other proteins, which is regulated by the genes. But then again, before we come to that gene expressions, we have all sorts of different input outside. It can be chemical input, it can be biological input, or it can be mechanical inputs. So the workflow is more like a circle actually, because This is not a single directional event. So it's not like extracellular matrix is applying a force on the cell and the cell is reacting to it. It's actually bidirectional. As much as the outside is applying force on the cells are also applying force. So it's yeah, it's like a circle going on there. And I think this fibrosis tissue mechanics is a good example for that, because your tissue starts to stiffen, that activates the cell, activated cell stiffens further the tissue, and it goes on like this in a circle, and eventually you lose your tissue. I don't know if it explains.
Okay, can we, in this case, take, no, how to say, compare the cells with micro robots, which is self-regulated by environment?
Yes, true. Well, yeah, I do not know much about the micro robots, but so I can imagine that's working in a similar principle, basically sensing their environment, and their movement is probably depending on what do they sense, I can imagine. Is it something like this?
Okay, thank you.
Excuse me, may I ask some question?
Yes, yes.
But please.
My first question, it is, can it be possible to use the biological processes in the each other biological processes, like a computer, the logical operation, like or, nor, or not?
Yes,
elements?
absolutely. Again, completely outside of my field, but I remember from my master's thesis when I was reading about these papers, DNA is actually perfect for logic processes. DNA you can design in however shape you want, and you can really control it such that it's just going to have this, like, how do you call it? One or zeros or on and off. But I know people do that. I know people are using biological DNA or RNA to have logic operations.
So where we use the DNA code, like a logical element, it can be like a super difficult supercomputer. What can remember the many terabytes of the files and information.
Absolutely, absolutely, yes.
And
no, it's true. Yeah.
Yeah, it's true. So what's the percent of the future we can use the not mechanical and electronic computers, but use the biological with the mechanical processes to control and know the some information and calculated some difficult mathematics functions.
I understand. So, of course, I cannot give you a percentage, but in just like last 10 years, there's an insane amount of improvement in nanobiology. And that is actually coming from the interest of not biologists or we people, but it's coming from the interest of physicists and the engineers. They want to use it, and I can really see that it's improving fast. Again, okay, if you are talking about these machines, can we use the biological machines more often? Cells, I don't think so, because as I explained, they're extremely heterogeneous beings. So you cannot really control a cellular behavior, as it doesn't matter how much you want it. But structural biological things such as DNA, yes, you can use it. And they are extremely stable, of course, in the environment too, like they can exist for millions of years. So at the end of the day, I think we need to put more research into it. Because we are at this state now, okay, DNA origami, we are making smiley faces with DNA origami. Yes, we can do many things, but again, you are not going to design it by hand writing it. So we need good models and good algorithms to be able to design the DNA as we want it. So yeah, I think in the future there will be absolutely more and more DNA-based machines and this kind of things that we are going to see, but I don't think it's happening so fast now.
Mm-hmm. And I interested about the next question. Can it can be possible to make artificial biological components to make the prostheses what can have no so big difference with you know the our day prothesis, what change
Yes.
the less arm or less leg to
Yes,
change
true.
him to mechanical. What can it can be make the with 3D just like 3D printed the muscles, bones, neurotic connection to make the
True.
another hands to operating the poor military or poor police officers what
Yes.
lost his eye or ear or finger or arm on the battlefield. It can be possible or not?
Well, first of all, I loved your questions. I think we can honestly discuss about these questions for hours. I love also talking about this like nanobiology and its implementations in the future. How much we can change in our body? Yes, indeed, we are using prothesis because it kind of replaces the bone, which is something that we don't really need much, not that much. Neurons depends, I think, if you are going to rely or believe in a kind of computer system that can process all that millions of thoughts in the same time, but it is possible. People, of course, as you might heard already, are trying to save every time an electron or like electric pulse is fired in a neuron, so that we can make a map of the interactions in our neurons, put it in a chip, and then transfer it to someone else, someone else's body. I think that is also possible. But then again, everything is possible, right? It's just, are we there yet? That's the question. So yeah, I don't know what to say, to be honest. I think, yes, for the eyes, all we need some sensors to be able to capture the light. Yeah, bones, as we said, proteins already there, already working. So for the ears, all we need hearing devices. You don't even need ears anymore. You can lose your hearing and you will still be able to hear with some sort of device. So it's true, we are moving there, but I do not think it's ever happening, because as much as some people like to have it, I'm pretty sure some people will be against it. And I don't think FDA doesn't even like approve super safe drugs. So I don't think FDA is ever going to approve any of these things, to be honest.
So thank you for your answer. Thank you for your answer.
Of course.
Please call it. Any questions?
Please, Samet, I would like to ask concerning the last slide, namely this very beautiful photo. This image, this is a fault colors or real colors? Because I see that
no, it's always a false color. No. So okay, so when we use the fluorescent microscopy, we are detecting the emitted light, and emitted light comes with a certain wavelength. It can be in the green wavelength, like 488. It can be red wavelength, 560, 650 wavelength. So detector detects that, but of course, we cannot see it because this is just like a single photon coming from somewhere, so we cannot detect it. But then what we do is to apply a false photon, especially in this case. So normally what we try to do is, if you have a green fluorescent protein, we assign a green color to that detector, detecting that, so that what we see on the screen is also green. But in this case, it's actually time-dependent experiment. As you can see, like they are taking one image at certain time point, and they simply assign a false color to that time point to see how the cell is spreading over time here.
Okay, thank you. And another question: you say that one of the idea of this method to find how the forces transform to another chemical stimulus which govern the behavior of the
Yes.
And please give some examples of what stimulus can be introduced by the forces which you measure.
Of course. I think the best example that I can give you is cell contractility or the cell motility. So okay, let's imagine the forces are created outside. The cell sense that it's stiff enough for it to move around, because if you are using very soft material, the cell is not going to move for sure. And then this information went all the way to the nuclear membrane. So in the nuclear membrane, as I mentioned, there's proteins called nesprins. These nesprins, upon a certain applied force, goes through conformational change. When it goes through conformational change, more other proteins can come and bind to it, actually. And when it does, these proteins determine how much the nuclear core pore complex is going to open. A nuclear pore determines which transcription factors enter and exit the nucleus. So let's say that also happens. And now we move inside of the nucleus. And inside of the nucleus, just like inside of the cell, we have signaling pathways. One is the very famous one is a YAP pathway. And that determines which contractility genes to be activated and which are to be activated. It can literally tell to the DNA, do not express this part of the DNA, express this part of the DNA. So depending on that, it can either make the cell completely shrink and even die, or it can make the cell spread around everywhere. So this is one of the outcomes that it can be, but it can be really many, many other things. Does it answer? I don't know if you have any question, please go ahead.
Okay, and what kind of the cells do you investigate? I mean, this or simply saying, do you investigate the neuron or synapses, something like that?
Synapses, we haven't started investigating it because focal adhesions are not really prominent there. And well, for the initial studies that we are doing now, it's just really, we are still at this stage that we are learning how can we work with these force sensors. To me, it took me 2 years. I swear, like now I start to get these images and I'm really happy with it. So in order to optimize this kind of new method, new technique, we need to work with model organisms. So for instance, if you want to work cell-cell junctions, your model organism can be endothelial cells. If you want to work on the focal adhesion forces, then your model organisms is mostly fibroblasts, because fibroblasts are very, very active cells. Neurons, in the case of neurons, the forces may not be that important because neurons are more like suspended in the brain. So they are actually not much of an applying forces. In fact, that's the idea, like, so that our brain is protected, so that the neurons are not disturbed, so they are not squeezed, or so and to the best of my knowledge, there might be not even a focal adhesion associated with the neurons. It may not be a thing there, but if you are interested in it, the forces in the nuclear membrane, of course, you can anytime measure it in the neurons too, because every cell has a nucleus and every nucleus has a nuclear membrane.
Did you estimate the?
I think we lost the connection for a second. Could you please repeat the question?
Okay. Did you estimate? Internet connection was lost.
Connection now is good, Gennady.
Yes. Now I was hearing it. Yeah.
Okay. Did you estimate the total power which produced by these forces by cells? Did you estimate?
By cells.
Yes.
Total one, I haven't, but it should be relatively easy to calculate because, okay, so in the case of force sensors, it's hard because, as I said, we like to stop at Fred efficiency. We do not talk about the values of the forces. We can estimate it. So what we can do, we can simply go ahead to this calibration curve, or just to simplify it, to this calibration curve. And imagine you are getting, let's say, 15% Fred efficiency. Then you can say that, okay, in theory, this should give you approximately 3 piconewton forces. Then what you need to do is to look at your intensity image. Intensity image is going to tell you how many vinculin proteins are there. So you need to calculate the total amount of vinculin. And for each vinculin, you need to also calculate what will be the estimated force. Then all you need to do is to sum up. It is relatively easier for TFM, even though it will not be accurate, because in the case of TFM, for instance, in this image, you already have a force field. So all you have to do is to take an integral of all these forces
Yes,
here, and then that will give you an approximate total force there.
Mm-hmm.
Maybe in fact it's even better for this, because in the case of traction force microscopy, you're actually looking at the total forces outside of the cell, which means that force is already applied outside. So by looking at outside, you can see how much force is there for sure. In the case of molecular sensors, it's a bit more tricky, because for a given pixel, as you know, our images in the microscopy comes with pixel. Within one pixel, there can be more than one vinculin protein, which of course then we cannot differentiate anymore because we are diffraction limited, which means we cannot know how many vinculin proteins per pixel.
Okay, thank you. Thank you.
Yeah.
Okay, we have a question from the chat. Maybe I read it. Okay, a question from Andrei Korolevich. Do you support the idea of transhumanism, not only for the treatment of diseases, but in general to improve human capabilities with the help of technology?
I think I will skip to answer this question. No, I'm going to give an answer. I do fully support it, because every technology, every medical technology that we have right now is coming from the people who supports this idea. So when you put a pacemaker in your heart, that is what is keeping you alive. You can die when you are in your 40s, or you can die when you are in your 80s, just because this very little tiny bit of device that is placed in your heart. People who lose their hearing, people who lose their sight, now we have options for this. So of course, these are medical approaches. I think your question is more like, yeah, do you prefer it just to make your body better? Yes, because if I'm in pain, I am taking an aspirin, right? And why not me never being in a pain? I mean, I would love the idea. And as someone getting as middle-aged nowadays, every morning waking up with a back pain, I think I would appreciate and I will support it. It's not that I'm going to work on it because I'm a molecular biologist. Obviously, I'm working on proteins and stuff like that. But in theory, I think it's an appealing one, let's say.
Okay, thank you. Any question, colleagues? Maybe I have a more
Please.
utilitarian question.
Ooh.
What software did you use to process the TFM images when you superimposed the perturbed and unperturbed pictures?
Sorry, can you repeat it again? The beginning of the question.
What software did you use?
Yes, it's MATLAB actually. So in
okay,
Leuven, yeah, it's MATLAB based, but it's now open software now, so you can actually just download it and use it. All you need to do is to give the cell image, give the stress state beat image, give this relaxed state, and software calculates it for you. So it's entirely written in MATLAB indeed.
Okay, thank you.
Is that it, I guess?
Okay, colleagues, any question?
Okay.
Good, as there are no more questions. Thank you, Sam, for your presentation.
Yeah, me thank you once again for patiently listen to me and ask very interesting questions. It was an honor for me to present this to you. So me thank you.
I wish you a doctoral degree.
I wish myself that too in a couple of years, hopefully. Okay, thank you so much then. Have a nice day to all of you.
I hope this will start.
I would like also
I hope this start and we will meet again on our seminars.
Okay, thank you very much. I would like also say thank you very much,
Of course,
Sam,
thank you for inviting me.
Very interesting and very attractive presentation. And I see really that there are a lot of things which could be very useful and very attractive for applied physics.
Indeed, absolutely.
Yes, especially concerning the calculating the stress, uniform stress near the cells, and probably more precise calculating the stress in the second method, threat method.
Yes.
Probably yes. And as you can see that it is really interesting topic in sense of future application of biological computing, but as far as I understand, this very interesting stepwise behavior of the DNK is useful for computers of von Neumann architecture, not for so-called non-von Neumann architecture. But I'm so sorry. This is just my
No, it's true. It's true. Yeah.
My thought. Thank you, and I suppose that it was very interesting also for our colleagues from Institute of Physiology. I see, so that some people were here, and have a nice day, have a nice holidays, and happy Christmas.
Soon, yes, soon, yes, it's true. True, thank you.
Probably
Okay.
too early, but anyway, thank you.
Yeah, thank you.
Thank you.
Okay,
Thank
bye-bye.
you. Okay, bye.